Hello, everybody, and welcome to the second class in neuroanesthesia titled Brain Metabolism, Autoregulation, and Reactivity. Brain metabolism. This involves planning complex behaviors, making decisions, and processing emotional and social contexts come with high energy requirements. The brain contains only 2% of the total body weight, but it accounts for 20% of an individual's energy expenditure at rest. This has been quoted by Ketty in 1957 and Sokolov in 1960. The neurons use 70 to 80% of the plan energy. The remaining portion is utilized by glial cells, that is astrocytes, oligodendrocytes, and microglia. Allocation of energy to maintenance, growth, reproduction, and higher cortical functions such as communication, imagination, and creativity. The brain metabolism can be split into two parts. That is the portion that drives the work of the brain, that is via synaptic transmission, that is activation metabolism, and the portion necessary for cellular integrity, which is via the basal metabolism. Basal metabolism can be defined as the maintenance of the normal transmembrane ionic gradients, that is keeping the potassium ions inside and the sodium and the calcium ions outside the cell. The remainder, the protein transmitter synthesis and other basic cellular functions. The neurons in the adult brain rely mostly on glucose as an energy source. And during prolonged fasting periods, ketone bodies are used during brain development. And in cases of intense physical activity, lactate utilization is increased. The brain depends on a continuous influx of substrates from the blood due to its high metabolic demands and negligible intrinsic energy stores. The exchanges of molecules between the blood and the cerebral fluids are regulated by the blood-brain barrier and the blood-cerebrospinal fluid barrier. This limits the free diffusion of solutes between the blood and the brain fluids. It selectively transports essential nutrients, ions, and signaling molecules. It removes metabolic waste products. It controls the influx of metabolites such as glucose, amino acids, and ketones from the blood into the brain. It prevents the access of blood-bound molecules and cells, example lymphocytes, that could be detrimental for neuronal functions. This is a diagram that depicts the basement membrane, pericytes, and the astrocyte perivascular end feet. The passive distribution is limited to gases and small nonpolar lipids. All other nutrients requires passive or active mediator transport. The GLUT1 to 5 transmembrane receptors transport the glucose via the cell membranes. And the monocarboxylic acid transporter 1 to 4 is depicted in the diagram. Large peptides and proteins such as hormones, growth factors, and neuroactive peptides are transferred via the receptor-mediated, absorptive-mediated, and carrier-mediated transport. Based on the requirement or not to hydrolyze ATP to move the solute across the membrane, there are two major families of transporters have been identified in the blood-brain barrier. That is the ATP binding cassette protein and the solute carrier proteins. The ABC transporters include the multi-drug resistance associated proteins such as MRPs and ABCB 1 to 6, P-glycoprotein, and breast cancer resistance protein. These act as efflux pumps that couple ATP hydrolysis to move the lipid-soluble molecules against the concentration gradient. The solute carrier proteins comprise a large superfamily of more than 300 members. They ensure a stable supply of carbohydrates, amino acids, and monocarboxylic acids, nucleotides, fatty acids, and organic anions and cations. Coming to the GLUT receptors. The GLUT1 mediates the intake of glucose into the brain. It has two isoforms, that is a 55 kilodalton isoform and the 45 kilodalton isoform. The 55 kilodalton isoforms is present in the endothelial cells of the blood-brain barrier. The 45 kilodalton isoform ensures delivery of glucose to the glia, ependymal cells, and the choroid plexus. The GLUT3 mediates uptake of glucose in the neurons, It is concentrated in the axons and the dendrites. The GLUT3 has a high glucose affinity and transport capacity. It ensures that the neurons receive a constant supply of glucose even when the interstitial glucose concentrations are low. The GLUT4 is insulin sensitive and it is present in astrocytes, neurons, and in endothelial cells. The GLUT4 is the GLUT8 is located in the cytoplasm of the neurons, mostly in the hippocampus, amygdala, cerebellum, and hypothalamus. The GLUT2 is a subset of the glutaminergic neurons in the hypothalamus. It is recently identified as a brain glucose sensor that triggers sugar-seeking behavior under hypoglycemic conditions. The MCT1 to 4, which is the monocarboxylic acid transporter, is present in the cells of the blood-brain barrier. It is responsible for bidirectional passive proton-linked transport of lactate ketone bodies, which are the acetate and 3-hydroxybutyrate and pyruvate. Monocarboxylic acid transporter 1 has a high affinity for pyruvate and also transports lactate and ketone bodies. It is present in endothelial cells, astrocytes, oligodendrocytes, and microglia. Monocarboxylic acid transporter 2 is a major transporter in neurons and it is concentrated in dendritic spines. MCT3 transports the lactate, retinal epithelium, and choroid plexus epithelium. MCT4 carries lactate and is only seen in astrocytes. The glucose metabolic pathways in neurons and astrocytes. The neurons are predominantly oxidative, where the astrocytes are mostly glycolytic. They are involved in the production of adenosine 5-triphosphate. It is also used to generate metabolic intermediates for the synthesis of fatty acids and other lipids required for membrane and myelin synthesis, the amino acids for protein synthesis and neurotransmitter production, and the 5-carbon sugars for the synthesis of nucleotides and to produce glycogen in astrocytes. In neurons, each molecule of glucose is oxidized via glycolysis, the pentose phosphate pathway, the tricarboxylic acid cycle, and oxidative phosphorylation with the production of carbon dioxide, water, and 30 to 36 molecules of ATP, depending upon the rates of proton leakage in the mitochondria. So these two diagrams describe the process of entry of glucose into the neuron and the astrocyte. Once glucose enters the neuron, hexokinase metabolizes it to glucose 6-phosphate. It gets further broken down into fructose 6-phosphate and fructose 1,6-phosphate, and finally to pyruvate. Pyruvate enters the mitochondria to enter into the tricarboxylic acid cycle in the form of acetyl coenzyme A, and finally ATP is produced. And in the same process, the glucose 6-phosphate can be converted to glycogen, and proteasomal degradation can also happen to the glucose 6-phosphate once it gets transformed to fructose 2,6-bisphosphate. And in the astrocyte, the glucose enters into the cell, and hexokinase breaks it down to catalyzes it to form glucose 6-phosphate. And the end product is pyruvate, which enters into the enters into the mitochondria to form the acetyl coenzyme A, and finally form ATP. And pyruvate in the astrocyte gets converted to lactate with the effect of LDH5. It gets transported to the MCT1 and MCT4 to form lactate in the interstitial space. And the lactate can also enter into the neuron via the MCT2. The glycolytic process metabolizes glucose to pyruvate, which can be actively transported into the mitochondria, where it is converted to acetyl coenzyme A. Acetyl coenzyme A is complexed with citrate, which undergoes a series of regenerative enzymatic reactions producing reduced nicotinamide adenine dinucleotide and flavin adenine dinucleotide in the TCA cycle. The NADH and FADH produced during glycolysis in the TCA are subsequently reoxidized in the electron transport chain. The electron transport chain utilizes the energy produced with the transfer of electrons through its various complexes to transport protons across the inner mitochondrial membrane into the intermembrane space. The flux of protons back into the mitochondrial matrix is mediated by enzyme ATP synthase. This utilizes the energy to generate ATP from ADP. Once inside the cell, glucose is converted to glucose 6-phosphate by hexokinase. Glucose 6-phosphate is then further metabolized via glycolysis of pentose phosphate pathway, or it can be used for glycogen synthesis, as I described. in the diagram. Glycogen is the largest energy reserve in the brain, although it is negligible in the peripheries. The enzymes which act on glycogen, the glycogen synthase and the glycogen phosphorylase. Glycogen is produced and stored exclusively in the astrocytes. In the neurons, glycogen synthase is maintained in a constitutively inactive state by hyperphosphorylation via the glycogen synthase kinase 3. Now, it can undergo ubiquitin-dependent proteasomal degradation mediated by the malin-laforin complex. And coming to the monocarboxylic acid metabolism, there is a clear evidence for a role of lactate in the brain metabolism in vivo, and this has been obtained very recently. So there is a cell type distribution of the monocarboxylic transporters and the intrinsic metabolic properties of the astrocytes and the neurons. So these have led us to believe that the lactate is shuttled between the two cell types to support neuronal metabolism. So under resting conditions, the lactate uptake by the brain provides about 8% of the energy requirements. And this percentage increases to about 20% under conditions of high plasma levels of lactate, such as intense exercise. Ketone bodies, these are recognized as an essential energy substrate for the brain during development, and it delivers around 30 to 70% of its energy requirement. The ketone bodies are the beta-hydroxybutyric acid and the acetoacetic acid. In the adult brain, the utilization of ketones is greatly reduced in the fed state. And during conditions of limited glucose availability, such as fasting, starvation, low carbohydrate, high fat intake, and prolonged intense exercise bouts, the complete oxidation of acetoacetic acid yields 23 molecules of ATP, whereas 3-hydroxybutyric acid generates 26 molecules of ATP. Now the astrocyte neuron lactate shuttle hypothesis. In this, you can see that the glutamate secreted by, released by the neuron enters into the astrocyte via the GLT1 transporter to form glutamine. The glutamine is converted to glucose via glycogen and then to pyruvate and lactate. And the lactate is transported out of cell via the monocarboxylic acid transporter via MCT1 and 2 and enters the neuron in the form of lactate. So the activation of nerve cells leads to the release of the neurotransmitter glutamate. The glutamate is actively taken up into the astrocytes with the glutamate transporters, GLT1, and is converted into glutamine. The uptake of glutamate into astrocytes stimulates both increased glucose uptake from surrounding capillaries via glucose transporters and increased aerobic glycolysis. Aerobic glycolysis can be stimulated by the breakdown in intracellular stores of glycogen. The pyruvate is converted to lactate by lactate dehydrogenase isoenzyme A, and it is transported out of the cell by the monocarboxylate transporter 1 or 4. It is then transported into the nerve cells via the monocarboxylic acid transporter 2. The lactate dehydrogenase isoenzyme B within the nerve cells converts lactate to pyruvate, which is used to fuel oxidative phosphorylation within the mitochondria. Glucose can also enter nerve cells via the GLUT3 transporters. This describes the ketone body oxidation. Coming to autoregulation. The lack of substrate reserve in the CNS and the inability to sustain anaerobic metabolism for more than a few minutes requires a constant blood flow, and this has to be finely tuned to the metabolic needs of the tissue. The metabolic rates differ considerably within the brain tissue. For example, there is a fourfold difference in the cerebral metabolic rate for oxygen between the gray matter and the white matter. Coming to the regulation of cerebral blood flow. The cerebral blood flow and the metabolism are said to be coupled, and under physiological conditions including sedation and general anesthesia, this coupling is generally preserved. So a rapid and precise regulatory mechanism has evolved in the CNS where instantaneous increase in metabolic demand can be rapidly met by a local increase in cerebral blood flow and substrate delivery. The term autoregulation has been used to describe the hemodynamic response of flow to changes in perfusion pressure independent of flow metabolism coupling. The cerebral blood pressure autoregulation. The process whereby the cerebral arteries maintain a constant blood flow in the face of changing cerebral perfusion pressure is referred to as cerebral blood pressure autoregulation. The cerebral blood flow is regulated by a complex system influenced by multiple factors such as cerebral perfusion pressure, brain metabolic activity, autonomic innervation, vasodilators, and drugs such as acetazolamide and papaverine. The relationship between cerebral perfusion pressure and cerebral blood flow, when regulated by intact cerebral autoregulation mechanism, is described in the diagram below, in which cerebral blood flow is effectively maintained between the perfusion pressure of about 60 to 160 millimeters of mercury. The cerebral blood flow is kept relatively constant at a wide range of the cerebral perfusion pressure, which is the driving force. Above and below the lower limits of autoregulation, the regulation mechanism is exhausted and the hyper-respective perfusion is possible. At normal tension, that is at a partial pressure of carbon dioxide between 20 to 80, there's a linear response of the cerebral blood flow. That is, the cerebral blood flow changes about 2 to 4% for each millimeter change in the partial pressure of carbon dioxide. As the partial pressure of carbon dioxide approaches the extremes, the linear response breaks down. Depending on the methods employed, whether it is a hemispheric or the cortical blood flow is measured, it is highly variable. Doubling the partial pressure of carbon dioxide from 40 to 80 millimeters of mercury doubles the cerebral blood flow. Halving the partial pressure of carbon dioxide from 40 to 20 millimeters of mercury halves the cerebral blood flow. Now, Arun Aslid, in his paper Cerebral Autoregulation and Vasomotor Reactivity, has put forth different hypotheses of autoregulation. For example, regulation can be metabolic, in which there is a balance between the cerebral metabolism, that is the demand, and the oxygen delivery, and it is mediated via a vasoactive substance which maintains the cerebral blood flow. And myogenic regulation, in which the effect of transmural blood pressure changes are directly detected by the vascular smooth muscle, maybe due to a stress sensing mechanism, and the calibers are adjusted accordingly to keep the blood flow constant. And in neurogenic regulation, the vascular smooth muscle in the resistant vessels are controlled via sympathetic innervation, and these receive inputs from the brainstem autonomous control center. So nitric oxide released by parasympathetic fibers may also play a role. The endothelium. The endothelium is the dynamic source of many vasomodulatory molecules. The mechanical receptor properties that contribute to cerebral autoregulation, which are the shear stress and the transmural pressure. An increased flow velocity, that shear stress, has been shown to induce vasoconstriction independent of transmural pressure. And an increase in the transmural pressure also causes vasoconstriction. Coming to the myogenic hypothesis. Baylis in 1902 described a myogenic hypothesis of cerebral autoregulation. There he focuses on the mechanoreceptor properties of the smooth muscle cells themselves. The stress-activated ion channels are identified to be non-selective cation channels. The membrane depolarization results in influx of calcium through the voltage-gated calcium channels and the smooth muscle constriction. Coming to flow metabolism coupling. This refers to the brain's ability to vary blood flow to match metabolic activity. An extensive branching of the perivascular nerves also serves to modulate cerebral blood flow, and which is known as neurogenic regulation. So there are two cell types which act as flow metabolism coupling, which are the endothelium and the astrocytes. The mechanism: potassium and hydrogen ions are produced by synaptic transmission. But the increase in these two ions stimulates vasodilatation, and this is a possible mechanism for neurovascular coupling. ATP sensitive potassium channels are found in vascular smooth muscle, and this has been implicated in the increased levels of potassium and hydrogen. Here there is a direct link between the neuronal activity and the cerebral blood flow. The phenomenon of carbon dioxide reactivity is mediated through the action of hydrogen ion on the cerebral arteries rather than the carbon dioxide itself. Coming to the metabolites, The byproducts of the neuronal activity, most notably in adenosine. The extracellular levels of adenosine rise sharply with neuronal activity, and the topical application of adenosine to cerebral microcirculation causes vasodilatation. So this is the neuronal regulation of cerebral blood flow, in which the neurogenic regulation has an extrinsic and intrinsic mechanism. It also depicts the microvascular communication and the vasodilatation caused by potassium and hydrogen ions, and the flow metabolism coupling seen in the endothelial cells. So coming to the neurogenic regulation of cerebral blood flow, the neurovascular unit is defined as a functional unit of endothelial cells, perivascular nerves, and astrocytes. It has the extrinsic and intrinsic perivascular innervation. And there are three major sources of extrinsic perivascular innervation which have been identified, which are the trigeminal ganglion, the superior cervical ganglion, and the sphenopenetrant ganglion. These ganglions carry sensory, sympathetic, and parasympathetic nerves respectively. Once the blood vessel dives deep into the parenchyma and leaves the virtual urban space, they lose the extrinsic innervation and the intrinsic innervation begins. So the majority of these nerves connect to the astrocyte foot processes. Depending on the area stimulated, there is an increase or decrease in the cerebral blood flow. Innervation of the cerebral microvasculature has been studied to be the nucleus basalis, the locus coeruleus, and the raphe nucleus. And there is a local interneurons which play a role in the regulation of the microvascular tone. And it has been recently noticed that the stimulation of the GABAergic interneurons causes vasodilatation of the regional microvessels. Coming to the endothelium, there are four major chemical systems which are the nitric oxide, endothelium-derived hyperpolarization factor, the eicosanoids, and the endotheliums. Furchgott and Ignero in 1988 discovered that nitric oxide is a diffusible second messenger that activates guanylate cyclase present in smooth muscle cells. The guanylate cyclase in turn synthesizes cyclic GMP, which causes smooth muscle relaxation through protein kinase G, and this causes activation of potassium channels and closure of voltage-gated calcium channels. Now, there are several isoforms of nitric oxide synthase, which is the endothelial nitric oxide synthase and the neuronal nitric oxide synthase, that is NNOS. There's another, which is the inducible nitric oxide synthase, which has been found in the brain under pathological conditions such as hypertension or exposure to endotoxin. Coming to the eicosanoids, are a group of vasoactive chemical mediators that are derived from arachidonic acid. So there are three main enzyme systems, which are the cyclooxygenase, lipoxygenase, and epoxygenase. The vasodilators prostacyclin and prostaglandin are predominant in the normal endothelium. The vasoconstrictive COX metabolites are presently thought to be the most relevant in pathological situations such as traumatic brain injury and subarachnoid hemorrhage. The ETA receptors are found predominantly in the vascular smooth muscles, and they are stimulated by ET1 and ET2 and mediate vasoconstriction. The ETB receptors are found predominantly within the endothelium and are stimulated by all three ligands and mediate vasodilatation. So in the presence of an intact endothelium, the ET1 binds to ETB and causes vasodilatation. And in patients which are stripped of endothelium, in vessels which are stripped of the endothelium, the ET1 binds to ETA and causes vasoconstriction. The astrocytes are also known to express a subtype of glutamate receptors that results in an increase in intracellular calcium. The astrocytic foot processes sense the synaptic activity and then responds to that activity by inducing vasodilatation of the appropriate blood vessels. Coming to another type of regulation, autoregulation, which is known as microvascular communication. It is been seen that there is evidence of communication within the blood vessels at the microvascular level, and this plays a key role in the overall regulation of blood flow in the brain. Rosenblum et al. First presented an in vivo evidence for the presence of coordinated vascular responses in the mouse bile arterioles. He found that local constriction response is initiated by micropipette administration of uridine triphosphate It can travel for more than 300 micrometers upstream from the point of application. Connexins, which is CX40 and CX43, these are present in the smooth muscle and endothelium, whereas CX45 has been seen in only seen in the smooth muscle. They have electrical and conductive properties that are different from one another. They have selective upregulation and downregulation of different connexins, which causes a change in vasomotor conductivity after an ischemic insult. Coming to cerebrovascular reactivity. This reflects the response of our blood-brain vessels to vasoactive stimuli such as neuronal activity. Cerebrovascular reactivity refers to the vasodilatory or vasoconstrictor response of the blood vessels to a stimulus, and this measure provides a direct assessment of vascular brain health. Impaired cerebrovascular reactivity indicates microvascular hemodynamic dysfunction, which is implicated in many brain disorders, including stroke and Alzheimer's disease, as well as mild cognitive impairment. The CVR index reflects the response of the cerebral blood vessels to extrinsic stimulation by vasodilators such as acetazolamide or hypercapnic challenge. Hypercapnia can be induced through inhalation of carbon dioxide enriched gas, which is a harmless way to investigate the effects of vascular functioning and blood flow parameters of the brain. So vasodilatation in response to carbon dioxide enhances cerebral reactivity through diameter enlargement in the small arteries, making it possible to evaluate the function of small intercerebral vasomotor vessels. So carbon dioxide is a powerful modulator of cerebral vascular reactivity. At one time, the carbon dioxide was thought to be the coupler between the flow and the metabolism. And because an increase in the metabolism generates carbon dioxide and releases the cerebral vasodilator into local environment. Carbon dioxide undergoes rapid diffusion across the blood-brain barrier, and this helps it to modulate extracellular fluid pH and affect the arteriolar resistance. Patient with hypocapnia may adversely affect cellular metabolism and shift the oxygen dissociation curve to the left. And a patient with severe hypocapnia, approximately 10 millimeters of mercury, can result in anaerobic glucose metabolism and lactate production. A decreased carbon dioxide reactivity can be a result of local decreases in cerebral perfusion pressure distal to a spastic or a stenotic vessel. This may reflect deranged metabolism or structural damage in a number of disease states, including head injury. Oxygen hypoxemia is a potent stimulus for arteriolar dilatation as a result of tissue hypoxia and concomitant lactic acidosis. So vasodilatation in response to hypoxia, it probably involves adenosine or potassium ATP channels. The cerebral blood flow begins to increase at a partial pressure of oxygen of about 50 millimeters of mercury and roughly doubles at a partial pressure of oxygen of about 30 millimeters of mercury. Hyperoxia decreases cerebral blood flow, producing a 10 to 15% decrease at 1 atmosphere. And a patient who's given hyperbaric oxygenation decreases cerebral blood flow, but high atmospheric pressure alone does not affect cerebral blood flow. Coming to the influence of age, the cerebral vascular contractility and dilatation becomes diminished in later life, and this impacts the neurovascular coupling. Multiple metabolic biochemical elements have been suggested as responsible for this loss of function, including the reduced production and release of vasodilatory substances such as nitric oxide, and increased production of vasoconstrictive factors by the cerebral endothelium. There are regional differences in the cerebrovascular reactivity. This is because reactivity in the brain is temporary dynamic and it differs between vascular territories and it is heterogeneous across areas. The research has shown that the regional cerebrovascular reactivity is higher in the parietal and the occipital areas when compared to the frontal, temporal, and the insular cortices. And the gray matter has 3 times greater reactivity than the white matter. Coming to the temperature. So cerebral metabolism decreases with the decrease in temperature. For each 1 degree centigrade decrease in temperature, the cerebral metabolic rate drops by approximately 7%. Another term called metabolic temperature coefficient, Q10, is defined as the ratio of CMRO2 at temperature T divided by the CMRO2 at the temperature that is 10 degrees lower. The value of cerebral Q10 in the physiologic range of 27 to 37% is between 2 to 3. Coming to the effect of anesthetic drugs, dose-related anesthetic or drug effects such as isoflurane, desflurane, and sevoflurane can alter the vasoactive responses just as the blood pressure and the carbon dioxide do. The vasodilatory effects of volatile anesthetics are apparent at minimal alveolar concentrations more than 1.5. Beyond 1.5 concentrations, the volatile anesthetics can blunt the carbon dioxide response, and it can render the cerebral blood flow pressure passive. In a patient with TIVA of propofol and remifentanil, it preserves the response to carbon dioxide and protects the pressure autoregulation better than that of volatile anesthetics. And because of the preserved flow metabolism coupling, increasing the depth of propofol anesthesia results in the decrease in the cerebral blood flow. Coming to the concept of a cerebral steal. So it refers to the decreased blood flow in the ischemic areas because of blood vessel dilatation in the non-ischemic areas. For example, it can be induced by hypercapnia. So blood can be diverted from one area to the other only when there is a pressure gradient between the two circulatory beds. And cerebral steal is said to be occurring in patients with cerebral arteriovenous malformations in which there is significant blood flow through the lesion and results in progressive focal neurological deficits. Coming to the Robin Hood effect. Supposing an ischemic area is maximally vasodilated, an increase in the carbon dioxide causes vasodilatation of the normal adjacent brain regions. This results in a decrease in flow. And supposing there is vasoconstriction in the normal brain, it causes the redistribution of blood to the ischemic region. This is known as an inverse Dillon or Robin Hood effect. So this mechanism can be seen in other cerebral vasodilators such as in volatile anesthetics, systemic vasodilators such as nitroprusside, hydralazine, and nitroglycerin. Thank you.